So what does that have to do with our body? Has a lot to do with our body because of the fact that when you go to the hospital in China, what is the first thing they do? They give you an IV, right? They give you an IV. Now, there's a couple reasons they do this. One, most people who live here are have very low amount of water in their body because they don't drink enough water, okay? So they're dehydrated. Second reason they do this is because there's not very much training for the pharmacists, and they can't trust that you'll get good medicine if they send you to a pharmacy. So they just as soon give you the medicine themselves, and the easiest way for them to give it to you is to give it to you in an IV. And the other thing is if they give you an IV, they get to charge you 125 kwai, right? Yeah. Yeah. So if you go get an IV, they are going to be adding fluid to your body. The chances of that fluid are actually going to be isotonic are very low. Okay? Frequently, there's going to be saline in there. There's going to be saline solution in there. But there's not going to be the same number of particles and molecules as you have inside your body. So, it's perfectly okay for them to do this, but what if they ended up giving you an IV for 3 or 4 days in a row? Then they'd be filling you up with fluid that didn't match your body, okay? So what would happen then is one of these two situations. These are blood cells, okay? So if they put you in a hypertonic solution, high solutes, okay? High solutes, if there's more solutes inside the blood cell, then the fluid is going to come in, right? There's going to be more water outside, and it's going to come in, and you're going to end up with a swollen cell that's going to burst. On the other side, if there's more water inside the cell, and the fluid has got lots of salt in it, okay, then the water is going to flow out of the cell, and it's going to flow into the blood, into the fluid part of the blood, and it's going to shrink. Okay? So that is why it's important to you, because in a situation in a hospital where you're given an IV, you have to be aware of the fact that you could give too much. So there's a really good chance that you're going to get a question about what will happen to the blood cell in isotonic and hypertonic solutions. So the next thing is facilitated diffusion. So facilitated diffusion is also considered, it's also considered passive transport, okay? Again, it's considered passive transport because there's no energy from the cell. That doesn't mean there's no energy, it just means there's no energy from the cell. So in this case, we're going to help. Facilitated means to help. If I gave you a facilitated take-home exam, that would mean you got help, okay? So in this case, what we're going to help is we're going to help things come into and out of the cell. If we're helping water in and out, it's called an aquaporin. An aquaporin is a hole. Because remember, the cell is actually hydrophobic. It doesn't like water. So if we're going to let water in and out, it's easier to do it with a pore. Or it can be a channel. A channel is going to let through an ion. It may be potassium or sodium or calcium or chloride, but it's going to let through an ion. And remember, those ions are actually surrounded by water. The last one is a transporter, and the transporter usually is going to help something like glucose or amino acids come into the cell. I've met this guy. He's really cool. And he received the Nobel Prize for showing about these aquaporins, and this was pretty recently. So the idea was they knew that the water went in and out of the cell, but they didn't know that it got help. And especially inside of the kidneys, these aquaporins are really important. Now, one of the things that happens when you get the Nobel Prize is they give you a ton of money. And so after he got his Nobel Prize and he got his ton of money, he decided he wasn't going to work on aquaporins anymore, that what he really wanted to work on was malaria, and he wanted to work on ways to cure malaria, and that's what he's doing now. Okay? Okay. So, facilitated diffusion through a channel. One, you're still working on a concentration gradient. Okay? That means you still have to leave an area with a high concentration to go to an area with a low concentration. Okay? Because diffusion is going to spread out, and all the little molecules are going to bump into each other. Eventually, they'll bump into some place where it'll shoot them into a channel. Okay? So that's why there's no energy from the cell, but there is, in a sense, an energy because of the fact that there's movement of the molecules. Okay? And there's a lot of pores, a lot of channels that will go through the membrane that are specific for only one kind. So a sodium channel, potassium channel, calcium channel, and these are all going to be really important in the next couple of days as we start to talk about the electrical impulses that go through your body, okay? Sometimes they have gates, and a gate will close or open a channel, okay? So just like, you know, you don't want to let all the sodium move into the cell. You want to make sure that you've got some control, so that's what a gate's for, okay? Okay. This is what is called a voltage-gated channel. The voltage-gated channel will actually move and change conformation in order to let the ions go through the center, okay? And it's going to let just a positive or just a negative charged ion go through, and the way it does that is by matching up the kinds of amino acids that have those charges on them. Okay? This one is called a ligand or ligand-gated channel. So any molecule that attaches to a receptor is a ligand, okay? It can be a big molecule, it can be a single amino acid, and we'll talk about that more tomorrow. But the idea is that something is going to make that open. And this one happens to be a sodium acetylcholine receptor, okay? So what it's showing you is that by something binding to the outside, it will open the channel and it will let the ion go through. So it's not electrically gated. It's not about positive and negative charges. It's about having a receptor that gets filled with it. It's a complementary key. This is a review of what I just told you, so you can read it when you're doing your studies about the kinds of passive transport, okay? Passive transport. The one thing that's not on here is the transporters themselves, and I think that's on the next. There we go. Membrane channels, upward and closed. Okay, transport. So a transporter often changes configuration, and that means that once the transport item is in there, it's going to actually change and open up. So here, once the transported item is in the middle, it's going to change so that it drops it into the inside. So it's either open to the outside or open to the inside of the cell, only one at a time, okay? So this would be, for example, something like glucose or amino acids. If you think about it, this is how the molecules that you have digested all the way down from chicken, down into proteins, down into amino acids. This is how it's going to get into the cells and into your body, okay? This is how the glucose is going to get into your body. It's going to get carried in by a transporter, okay? And then it's really in your body. When it's in your stomach, it's not really in your body, okay? It's just passing through. But once it gets absorbed, then it's in. And this is how that action takes place, is from these kind of transporters, okay? Okay. When we start talking about active transport, we can use energy to go against the concentration gradient, okay? The energy inside the cell will help us push out things that there's too much in there, okay? So whether it's sodium or potassium or one of the other ions, we can push it out using the energy inside the cell. So the most important part is to remember that it's got to involve the energy. And the energy inside a cell is usually in the form of ATP. Normally they're going to be called pumps, or they're going to be called, sometimes they're called ATPase, which means that they use the ATP in order to make the conformational change. Okay, so this is a pump, and you can see that it is taking the calcium and then using the ATP in order to push it out of the cell to the outside. Okay? So ATP is the energy mechanism. The ATP has a bond for phosphate, and when it breaks the bond, it actually causes the energy to go from the molecule into the pump itself and push the pump. So it actually changes the conformation. This one is the sodium-potassium ATPase. It's in all your cells, and it's essential that it work. When it doesn't work, you die. There's just no doubt about it. When it doesn't work, you die. So this one has got to exchange what's inside the cell, which is the potassium, which is the sodium, and it's got to pump it back out because we really don't want the sodium inside the cell. The inside of the cell is the domain for the potassium. So we've got to switch that out. So obviously the sodium got in here somehow. We've got to take it back out. So that's what this pump is showing you, is that it's going to take out 3 sodiums, and it's going, 3 sodiums, and it's going to let in 2 potassiums. Okay? Okay. Secondary active transport. Active means it's going to have energy from the cell. Secondary means that it's kind of using a roundabout way of doing this. Okay? Secondary active transport. Indirect energy consumption. Okay? You see that? Indirect energy consumption. And what it's doing is using the concentration gradient of one molecule to move another molecule. Okay? So, we know there's a lot of sodium outside the cell. The extracellular environment has a lot of sodium. The concentration gradient would be to move it into the cell, into the low sodium environment. Okay? And since we know that, we're going to use that to carry along something else. And in this case, it shows you that there's a number of these things that work together. There's sodium glucose. It's going to carry the glucose into the cell with the sodium. That is how you get the glucose out of your intestines and into your body. Okay? Sodium-calcium exchanger. An exchanger means that you're going to have to trade off. Okay? We use this a lot in muscles. We need to have the calcium so we can make the muscles contract, but they're going to stay contracted unless we take take the calcium back away. Okay? So we got to move it in and out. Sodium-calcium exchanger. Sodium-potassium 2-chloride symporter. Symporter means they all move in the same direction. And we use this one in the kidney so that we can move out the ions that we want to keep. We process a lot of blood every day, but we don't want to lose all of our ions. We've got to actually keep the ions in the body and just let the water and the waste products pass through. And then this one is a sodium-hydrogen exchanger, which we also use in the kidneys and several other places, including your brain. Okay? Hydrogen is going to help keep our pH in the right place. We all know that the human body is like 7.5 pH. We don't want it to go too high or too low. We got to keep homeostasis, so we have to keep control of the hydrogen. Okay. There's some other forms of transport through the cell that are not called active or passive transport. Okay? One of those is transport by vesicles. Okay? Vesicles can do 2 things. They can take things into a cell. They can also let stuff out of a cell. Okay? So, if there's something in the extracellular environment and we need it and we want it, then we can pull it in. So phagocytosis is the process of taking a large particle into the cell, okay? Because it's going to engulf it and it's going to keep it, okay? Amoeba do this all the time. So we're just, evolutionarily using the same thing because if it works well, why change it? Okay. Penocytosis is when we are actually just taking in fluid, okay? So it's just fluid and probably some small solutes that are inside of the extracellular fluid, and so the cell's just going to pinch off a little tiny bit full of fluid. So there isn't any big proteins or anything in there, okay? And then, of course, the other thing is we can also do exocytosis, and we can take things out, okay? So, for example, if you have a cell that makes a hormone, or it makes a cytokine, which is a messenger that's going to go to other cells, then it's going to send it out by exocytosis, by fusing with the membrane and letting those go out into the extracellular fluid. Okay. So this one gives you the examples so that you can see the endocytosis where we're going to take it in, and the exocytosis where we're going to let it go out. This is also one way that we can make our cell bigger and smaller, because in each case we are going to either fuse the vesicle and it's going to become part of the membrane, or we're going to pinch something off and it's going to make the membrane smaller. Okay? Okay. So this is what the real picture looks like. Over here on this side, these are the microscope pictures where you can see what it actually looks like. And in this case, it's about a receptor on the membrane. So the receptor is going to have its ligand. It's going to bind to it, and then it's going to get pinched off, and they're going to be internalized. So one of the ways that your body keeps having the messengers from being always active is by internalizing them and taking them away from the membrane. So if you don't need to be taking in any sodium channels, and your body can go ahead and take those sodium channels and make them inactive. Exocytosis, that's when we're going to be releasing the things that we've made inside the cell. Okay? And, okay. Make sure that you got this. Role of active transport processes. We've already got the pumps. Remember, it requires energy from the cell. Okay. There's a little summary for you, so when you're studying, you'll remember what each one of these means. Okay? Is it dependent on a sodium gradient? If it's going to be secondary active transport, the answer is yes. Remember that sodium is going to be the part that's going down its concentration gradient. Okay. One of the things about the transport is it has what we call saturation kinetics. Okay? That means that we can actually fill it to a max. Do you remember the picture where there were the receptors, and each receptor bound a ligand, and then it formed a vesicle and it went internalized? Okay, so you have a maximum based on the number of receptors there. Okay? If you think about diffusion, there's no maximum with diffusion. Okay? Anytime you start to use a transporter, you have a maximum. There's only so much that can go through the transporter. If you have a channel, depends on the size of the channel, but there's only so much that can go through it. So in those cases, you actually have a maximum. You have a maximum that can be transported, okay? And that becomes important in a situation where, say, for example, you have a drug, and it's going to kill a virus, and the virus is inside the cell. In order to kill the virus inside the cell, you have to have a certain amount of the drug inside the cell. Now, if that drug gets carried in on a transporter, what's going to happen if you fill up all the transporters and there's still not enough inside the cell? Then your drug's not going to work. Okay? Okay. So this kind of has a summary on it that shows you the sodium ATPase at the top and how it works with the ATP and what goes in and out. Shows you the channels, okay, and which direction they go. and also shows you the antiporter or the transporter where one thing goes in each direction. It kind of gives you a summary to give you an idea of what the possibilities are for the transport. Okay. So I have one more little movie for you. This one's been trying to play all day. Let's see if it actually...
The smallest unit of life is the cell. In order for life to be sustained, nutrients and gases must be able to pass into a cell. Wastes and other products must be able to pass out of the cell. Many of the materials that pass in or out of the cell do so by passive transport. In this process, there is no energy, no ATP expenditure by the cell. Concentration gradients allow for the movement of materials. Osmosis and dialysis, both specific types of passive transport, use this concentration gradient of high to low. Another type of passive transport is facilitated diffusion. Since it is passive, the high to low concentration gradient is present. However, the process is facilitated by a carrier protein. The carrier protein is somewhat like a revolving door, allowing easy access across the membrane and then rotating back to the other side. All of these processes, osmosis, dialysis, and facilitated diffusion, are passive. The cell does not have to use any energy to move materials by these methods. The concentration gradient is used. However, there are materials that need to be moved against their concentration gradient. This process requires energy, ATP, and protein carriers that are found in the plasma membrane. Defined active transport is material moved from a low concentration to a high concentration through a membrane using a protein carrier that requires ATP. An example of active transport is the mechanism of uptake of iodine by the thyroid gland. Iodine is essential for the manufacture of hormones that influence body metabolism. When you eat a meal containing iodine, the iodine will pass from the small intestine into the circulatory system. As the capillaries pass through the thyroid gland, iodine diffuses out of the circulatory system. Iodine attaches to protein carriers in the thyroid cell's plasma membrane. Being proteins, they have a unique shape, so only iodine will fit. Glucose, which is also actively transported into the cell, relies on a different protein receptor. The energy required is somewhat analogous to a rock being pushed uphill. The processes of simple diffusion, osmosis and dialysis, facilitated diffusion, and active transport allow for most all of the small material, those dissolved in plasma and extracellular fluid, to pass in or out of the cell. Large particles, however, pass into the cell by endocytosis. This is an active process. It needs ATP. In this process, the plasma membrane surrounds the particle, such as bacteria or a food morsel, and forms a vesicle that pinches off from the membrane. The cell does this in two different ways. One way is called phagocytosis. In this method of cell eating, large particles are surrounded when the cell extends the plasma membrane and engulfs the particle. The result is a membrane-bound package of material. Fluids that the cell wants to take in enter by a process called pinocytosis. In this type of endocytosis, the cell membrane sinks in, the material sinks inward, and the membrane pinches off. The cell uses energy for this movement. In both types of endocytosis, the cell...
Okay. So I didn't cover everything, but that was that... Okay. I think that is the end of that, so we have just one thing left, and that is your assignment that you're going to turn in next Thursday. Okay. So, this is going to be your very first assignment. It's going to be really short. Chicken pox. How many of you know that there was an outbreak of chicken pox here last year? Did anybody get chickenpox last year? It is so rare in the United States for somebody to have chickenpox in college that the first student who sent me in the email that said, I'm so sorry, I cannot take the test, blah, 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 I have the chickenpox, I thought they were lying. I'm sorry. So then I found out that really and truly we did have actually chickenpox outbreak on campus. So this is what I want you to do. And I wanted to mention to you that WebMD and the Mayo Clinic both have got really nice websites with lots of information. If you don't use Wikipedia or if you have some other site, that's fine. I want you to find out what causes chickenpox. I want you to know how it's spread. This is really important because you could be next. I want you to know how it's prevented. Okay? Can you prevent chickenpox? That's a good question for, you can put a yes or a no and tell me. Okay, so you've only got 1 or 2 sentences for each question. I am not asking you to give me an essay pepper. I don't want to read an essay pepper. I want you to just give me a couple sentences, okay? Please don't forget your name and your roll number so that I can give you credit, okay? So next Thursday when you turn that in, we will talk about how to prevent getting the chickenpox. Tomorrow we are in this classroom at 2 o'clock, okay? Tomorrow we will talk about second messenger systems, and we're going to talk a little bit about cholera, and we're going to talk about stress. We're going to talk about stress and cholera and how that relates to your cells, okay?